Welcome. This talk surveys 700 million years of apoptotic control centered on the BCL2 family, tracing how a conserved protein fold underlies cell death regulation across animals. Over the next slides, we'll move from broad evolutionary patterns to molecular structure, gene organization, functional assays, and therapeutic implications, keeping the narrative focused on how conservation and change shape apoptosis. This slide shows how different lineages sculpted the BCL2 family in distinct ways. Teleost fish expanded the toolkit through extensive duplications for diverse functions. Some eukaryotes lost the entire toolkit, and mammals retained a balanced core set of proteins. These contrasting paths illustrate evolutionary trade-offs between expansion, loss, and functional replacement over deep time. The BCL2 fold is an alpha helical architecture built from BH4, BH3, BH1, and BH2 motifs that together create a hydrophobic groove. That groove is essential for ligand binding and membrane insertion, and remarkably, it's been preserved from simple animals to humans, explaining why the same structural principles control apoptosis across metazoans. Here we highlight a gene structure lock preserved for 700 million years. Coding exons consistently terminate at a conserved GGW motif, separating the membrane anchor exon. Seeing the same exon architecture in sponges and mammals shows that gene organization has constrained and protected protein structure throughout evolutionary history. This panel presents an ancient molecular switch. Anti-apoptotic members sequester pro-apoptotic BH3 helices to control mitochondrial outer membrane permeabilization. From Trichoplex to Hydra to humans, the binding groove and sequestration mechanism are essentially identical, demonstrating that the core anti-apoptotic strategy is ancient, conserved, and functionally central to metazoan cell death control. Glycozoan BH3 peptides can engage human BCL2 proteins and sensitize cancer cells to chemotherapy, showing surprising cross-species activity. Structural analysis reveals 5 conserved atomic anchor points in the groove that support ancient recognition, while variable regions enable modern tunability, suggesting basal peptides are both evolutionarily informative and potentially useful as novel therapeutic scaffolds. This slide outlines the early metazoan BCL2 network, a tripartite architecture of effectors, guardians, and sensitizers that existed in basal animals like cnidarians. Expression data and inferred components show the full network was present and conserved, implying strong structural and functional continuity of apoptotic regulation across 700 million years. C. elegans illustrates a minimalist apoptotic control, a streamlined pathway with one sensitizer, EGL1, and one guardian, CED9, that produces efficient developmental cell death while lacking the mitochondrial permeabilization seen in vertebrates. This example shows how networks can be reduced yet remain functional, contrasting worm simplicity with mammalian complexity. This slide presents therapeutic opportunities from evolutionary mining. The conserved BH3 binding pocket is the current drug target, but mining basal homologs can reveal new scaffolds that bypass vertebrate resistance mutations. Exploiting evolutionary divergence offers novel mechanisms of action, reduced resistance risk, and promising new avenues for drug discovery and apoptosis induction. Despite an immutable BCL2 core fold conserved across metazoans, lineages remodel interaction partners, subcellular localizations, and redundancy levels. In other words, the fold is indispensable while the surrounding toolkit is evolvable, proving that apoptotic control combines a stable structural core with adaptable regulatory networks across evolutionary time. This slide focuses on investigating complete BCL2 loss in select groups, such as ctenophores, plecozoan isolates, and minor nematodes. Systematic surveys aim to correlate complete loss with alternative cell death mechanisms or unrecognized compensatory genes, helping us understand how essential functions can be replaced or rerouted in different animal lineages. Finally, evolutionary reconstruction and structural biology together guide synthetic biology. Ancestral sequence reconstruction reveals primordial pro-survival or pro-death activities. Structural integration explains how functions were acquired, and those insights enable design of custom cell death switches and synthetic approaches that harness ancient mechanisms for modern biotechnology.